My name is Wendy Gibble. I work at the University of Washington Botanic Gardens, and I live and work on the traditional lands of the Coast Salish people, which touches the shared waters of the tribes and bands of the Suquamish, Tulalip, and Muckleshoot nations. I really appreciate the opportunity to participate in this meeting and share information about our seed conservation program. So for my talk, I'm going to give you an introduction to our ex situ seed bank and present how we operate the seed bank, which is called the Miller Seed Vault. So with that, let me share the screen. And can you see that? Great. All right, so where we're headed is... So this is our seed vault. I thought we'd just start with something that you can put up an image to what we are talking about. It is located on the UW University of Washington campus, Seattle campus, in our Botanic Garden. It was built in 2003, and we got a grant from a nonprofit charitable foundation to build the seed vault, and we manage the collection. So the important thing to keep in mind with our seed bank is that it is a conservation collection, and the purpose is to secure germplasm of imperilled and culturally important species in a secure off-site facility. Ex situ means off-site, to safeguard against extinction and to preserve unique genetic adaptations. So for many seed banks, the focus is on conservation of rare species, because these are the species that are most vulnerable to decline and extinction. So as such, our focus is really very long-term storage, more than 20 years. And that really dictates how our seed bank is operated. And that's pretty different from seed banks that are operated for restoration purposes, where the storage is for storage, the focus is for storage for shorter periods of time and bigger quantities of material. So key components of the conservation seed bank that you want to keep in mind is that we are focused on very long-term storage, and we're really looking to maximize the genetic diversity of the populations and the species that are held in the seed bank. We want to bank just enough seeds to safeguard these populations, the genetic diversity, and to provide opportunities and flexibility for effective management activities. We also want to think about how to conserve the genetic local adaptations and what collections we make and what collections we keep banked to maximize our representation while minimizing artificial selection. In other words, as we put plant seeds into storage or as we propagate them, we will always be inadvertently selecting for certain lines, and we want to try to minimize that. And we also want to protect against genetic drift, where we lose certain adaptations or genes or alleles, or we in fact fix them where they are overrepresented compared to wild populations. So key components of seed bank for conservation, of course, is the cold storage facility. And you're looking here at our processing room in the seed vault, and our cold storage is done in just a commercial grade freezer. You need to have equipment to dry the seeds, and our entire processing room and our short-term storage room, which is to the right, is 15 degrees Celsius and about 32% relative humidity. And those conditions bring the water content of the seed down to 6 to 8%, really low. And so this is based on research that's been done on how to maximize shelf life of seeds. We have a rigorous data management system, which I'll touch on tracking these collections. And we have agreements to back up our collection. So most of the collections that come in, as long as it's in accordance with our agreement with whoever's providing the seeds, we do back them up in the national lab at in Fort Collins, Colorado. And then we do viability testing, not only to document the viability of the collection, but also to figure out how to propagate these species, since we're working with rare species that have never been propagated. An important piece of the puzzle is how to actually get them to germinate if we need to reestablish wild populations. And then all seed banks should have a distribution policy. What are the seeds available for, and what is the process that we would undertake to distribute them? So one of the most important considerations that you need to think about when making a seed collection and storing seeds is whether your seeds are desiccation tolerant. And seeds that can tolerate desiccation are called orthodox seeds. And these seeds are really easy to store long-term, and we can get very long shelf life with these seeds. And because they can tolerate desiccation, we can dry them out, like I mentioned in the previous slide, and then we can put them into very cold storage at about -18 Celsius. And that will give us a shelf life for many species that extends decades or longer. We don't honestly know how long some of these seeds will persist at those conditions. It could be centuries. Most of our temperate dryland species are orthodox, so we're very fortunate that this works for a vast majority of the species in our state. But some species do not tolerate desiccation, and we call those recalcitrant seeds. And recalcitrant seeds are require special handling to try to extend their shelf life. Typically, they would be killed. They just don't tolerate any kind of drying. And of course, if you freeze seeds without drying them, the water expands and kills the embryos. So these seeds can't be stored in a conventional freezer, and we would have to look at cryopreservation for these species, in which we're figuring out the chemical properties and processes in those unique species, and then determining how to best store them in liquid nitrogen and whether that can even be done. So it's a much more complex process and requires a lot of research. And we don't have liquid nitrogen at the University of Washington campus. We're storing with a conventional freezer. But if we were to store recalcitrant seeds, then we would be working with our national lab. And I just want to point out that this liquid nitrogen tank here on the right is at the Fort Collins facility. They have, that's a Department of Agriculture facility. They have about 40 of these, and this is where the United States food supply is backed up. And then this is Chris Walters in this picture, and she has done a tremendous amount of work on understanding the chemistry, biochemistry for seed storage. And so she's a great resource that we rely on. And then I'll also mention that intermediate seeds are seeds that are, you can get a short shelf life of maybe 2 to 5 years. They can tolerate some lower humidity, lower water content, but still can't be frozen. One thing to think about is that while most of our species are orthodox, not all of them are. So for instance, we do have some species in temperate rainforest that are recalcitrant. And we also will see recalcitrance maybe in some aquatic species. We see intermediate seeds in some of our hardwood trees. And then some salix species actually have intermediate seeds. So it pops up in interesting different lineages, and it's not always predictable. So I just wanted to touch on sources of seed collections that are coming into the Miller Seed Vault. As Karina mentioned, we have a big network of volunteers who we have monitoring rare plant populations across Washington state. And they help, and some of them will also follow up and conduct seed collections for our priority species. We also do seed collections ourselves, our staff, and then we have agency partners and other nonprofit partners contribute to the seed vault. This is, I don't want to get too much into the weeds on a spreadsheet because what could be more boring, but this is just an example of the type of tracking that we do with each collection that comes in. We are curating these seeds from the moment they are received to the moment that we distribute them out or replace them or use them for propagation. So for every collection, we're tracking information such as who made the collection, the date of the collection, where it was collected, who's the owner of the land or who manages the land where the collection was made, and their contact information, detailed information on the location, including a precise map, and then number of plants sampled, how big the population is, how many maternal lines are represented in each collection, and so on. And then we're also assessing seed viability, and we're tracking that all by each collection. So let me just briefly touch on the process for that happens when we receive a collection. This is on the upper left corner, a picture of a collection that was made by me. We like to use coin envelopes, and we prefer that all collections come in along maternal lines, meaning every packet holds seeds from one individual plant. And this allows us to track the genetic lineage of all our seeds in our collection. They get an accession number, like a museum record, that we can then track these seeds. And then we have volunteers and staff work in the seed vault to clean seeds. And it's we're working with small collections of typically a few thousand seeds. So this is a meticulous process that is done by hand. And we're also assessing seed quality as we do that. And we're removing chaff to try to keep the volume very low. We will create curatorial packages for our collections. And so those will be seeds that we'll use for doing germination viability testing. And then we split the collection in half and send half to the national lab and package the seeds by maternal line into these little glassine envelopes. And then we seal them in these special aluminum foil packets to they're airtight so we can preserve the dry conditions of the drying room before we put them into the seed bank. So our seed bank, which is that commercial grade freezer I showed you in an earlier picture, holds over 1 million seeds of about 149 native plants. This is really inexpensive to do. And you can get a shelf life of decades. And so we've always felt like, why wouldn't you do this? This is the safeguard you need to protect these precious species. I'm not going to go into depth on our viability and germination testing, but we do test every collection that has more than about 200 seeds. And we have these growth chambers in which we can program light and temperature to figure out how to break dormancy if that's needed. And I just wanted to wrap up with our distribution policy. So our seed bank is a conservation seed bank. They're not, the seeds are really only for conservation of wild populations. So we're not going to provide them for sale or for horticultural use. We support activities like introductions at appropriate sites and research that is going to help with plant conservation. So we also always work with the landowner or agency from where the seeds originated. and always would provide the seeds back to them upon request. And if a third party does ask us for some seeds, say for research, we then reach out to the original landowner or manager of these lands and say, hey, we've got this request. Here's the use, here's the proposal. Is this something that you'd be supportive of for providing seeds? So it's we're very careful about honoring sort of the source of these seeds. They're not for our use. So we're really just a repository to hold them for other people. And so I want to thank you, and I'm happy to answer questions. And I wanted to highlight this book called Ex Situ Plant Conservation, which is a really important guide and resource for the science behind seed banking. And with that, I will stop sharing my screen and happy to take questions.